selective reabsorption in the kidney. Explain how the kidney reabsorbs useful substances in selective reabsorption. So this is reabsorption. That means taking back into the blood. Okay, taking things back into the blood. And it's selective, so that means not all. So where's it taking things back into the blood from? Well, this is from the filtrate. The filtrate, the stuff that was filtered through the tiny slits, through the fenestrations of the capillaries and through the filtration slits of the podocytes in Bowman's capsule. So we created filtrate, which has got a lot of things in there that we want to take back into the blood. And so some of those things that we want to take back into the blood are glucose, because that can be used in respiration, salts, small proteins, also amino acids, and water. So all of these things are making up the filtrate. And so the filtrate has left the renal capsule and it's starting to go down into the proximal convoluted tubule. Okay, so here we are, and we are taking a section. So this whole diagram is showing us a section of this convoluted tubule, which is then going to run down into the loop of Henle. So we're kind of zooming in onto this tubule. Now surrounding the tubule will also be blood vessels. So blood vessels will be running close to this convoluted tubule and all the other bits, so that we can get absorption into the blood from the proximal convoluted tubule. So the blood vessel we can see on this diagram over here. So this is the blood vessel, and the proximal convoluted tubule is this part here. And so we're seeing only one side of it. So actually there would be another side which is the same as above that. And so the tubule is this part here, and through there the filtrate is running. So the filtrate had all those things that we talked about in it: glucose, water, salts, small proteins, urea, all those things. And so we want to now reabsorb through into the cells which are lining the proximal convoluted tubule and then into the blood. Now we could have a reasoned guess at how these things are going to get through. Glucose, which is C6H12O6, is large and polar. So because it's large and polar, it's going to need a way of getting transported. And that way of transport, in this case, we can sum up. It's going to move through via active transport. So there's active transport of glucose. We also have amino acids. And because they have a charge with So our amino acid structure. Because of this structure and the fact that this will go around to here and give us a positive group and leave this side being negative, it's large enough and charged enough that it's going to be slow moving through the membrane without a way to get through. So this is going to again be active transport of amino acids. We also have as well in here mineral ions. And those mineral ions can also be transported through active transport. So those are the three active transport things. So in terms of how much we're going to reabsorb, well, we would want to absorb almost all the glucose, almost all the amino acids, and most of the mineral ions, so almost all of that. Flowing through here, we've also got H2O, we've got water, and that's going to move in via the only way it can move ever, osmosis. So water, and most of it is actually absorbed here, so it's 85%. 80 to 85% of the water is absorbed here. So we get water absorption, and that's just going down the concentration gradient via osmosis. Now we also get, and this is kind of a 7-level statement, we also get some urea going back in. Now remember, we wanted to get rid of urea because it's a toxic waste product. But some of it will get reabsorbed, and that's kind of a high-level statement. And then also we said that we had some kind of small proteins. Now these small proteins are too big, they are large and polar. Or large enough and polar. We said they were small, but they're still large enough to not really travel through. So what these are going to do is they're going to go into a pit. We're going to get intake through endocytosis. And because it's actually in a liquid form, it could be termed, and this is a very, very much a 7-level statement, it's pinocytosis. And when they get in there, these will then be broken down into amino acids. And those amino acids, like everything else, like the glucose that we talked about, the water, the mineral ions, it's going to go back into the blood, and then that's going to go back to the heart and get pumped around the body again. So what's left then? We absorbed out lots of things. Well, still left is water. Still left are some salts, because not all of the salts were reabsorbed. Still left is a higher concentration of urea, and now, because we've taken water out as well, we're going to increase the concentration of the toxin, or any toxins that were there. So it increased. Our water level had decreased, but it's still there, because we had absorbed water. So the final question is, what is special about these cells? The cells lining the proximal convoluted tubule. Tubule just because it's got a hole in it. And this is, we could say that down here is the lumen. It's the hole in the proximal convoluted tubule. So what's special about these? What special features would a cell want to have if it's doing a lot of absorption involving active transport? Well, we can see there we've got lots of mitochondria. because that's going to lead to more ATP, and we need ATP for active transport. The other thing that we could see here, we see these folds, just like we saw in the small intestine. We see microvilli and they're going to increase surface area for faster absorption. Okay, so we'll get some marks as well for talking about the special features of the cells of the proximal convoluted tubule. Okay, so here's our quick recap. We talked about the proximal convoluted tubule having a microvilli cell border to increase surface area, and that means more absorption from the filtrate. Having lots of mitochondria for extra ATP because of the active transport. And we said that there were three things: glucose, amino acids, and mineral ions, which were being absorbed via active transport from the filtrate into the tissue fluid. It then diffuses in the end into the blood capillaries. So the tissue fluid was the small gap. If we look up here, the tissue fluid is this section here. It's this area between, because between a capillary and a cell is tissue fluid. So we've got small proteins absorbed by endocytosis, in particular pinocytosis, digested into amino acids and back into the blood. 80% of the water via osmosis, down the concentration gradient. And then these high-level statements that not all the urea is actually reabsorbed.